Hi everyone, a very good morning to all the participants and hearty welcome to the today's Pharmacy Education Webinar Series 8 being organized by Baid Metha College of Pharmacy in association with MYBO Group. So without any further delay, let me take the opportunity to invite Dr. C.N. Nalini Madam, Professor and Head Department of Pharmaceutical Sciences, Pharmaceutical Analysis Specialization, Baid Metha College of Pharmacy. Along with us, Professor Dr. Grace Ratnam, who is Principal of Pharmacy College. And now I request Dr. C.N. Nalini ma'am to please give the welcome note.
Good morning, everyone. I am Dr. Nalini, Professor and Head of Department of Pharmaceutical Analysis, Baid Metha College of Pharmacy, Chennai. Respected Principal Dr. Grace Ratna, distinguished speakers Mr. Ravi Jayapal and Professor J. Vijaya Ratna, Mr. Ishwar of MYBO Group, and dear delegates. On behalf of the management, principal, and faculty, it's a privilege for me to welcome you all. Baid Metha College is a pioneer in self-financing pharmacy colleges of Tamil Nadu. It functions with the motto of no capitation fee or donation. The college has achieved excellence in pharmacy education with the help of its well-qualified faculty under the able guidance of Principal Dr. Grace Ratna. Today's webinar is a webinar series 8 conducted by Baid Metha College of Pharmacy under the theme Advancing Scientific Knowledge in Times of Pandemic. Webinars are knowledge sharing platforms during the time of this kind of pandemic, and it's a great pleasure to greet all the delegates who have joined the webinar with a thirst for knowledge. I hope that the knowledge sharing by the two eminent speakers with vast experience will definitely fulfill the delegates' desire. On this occasion, I would like to extend my gratitude towards our principal for encouraging us to conduct a webinar like this today. Thank you, madam, for your kind support. Today, we all have gathered here to discuss about the important topics on impurities and variance analysis. Impurity profiling is an expanding topic in pharma industry, and it is also playing a significant role in academic research activities. No research is complete without statistics. In that way, our second topic, analysis of variance in research and DOE, becomes important. So, we have assembled here to know more about the topics. I take this opportunity to welcome the speakers once again, and I also welcome all the delegates who have joined this webinar from various colleges across the country. On behalf of our college, I extend my warm welcome to you all once again. Thank you.
Thank you very much ma'am for the welcome note. Now I take the opportunity to invite Dr. S. Amudalakshmi, Associate Professor at Baid Metha College of Pharmacy, to give introduction for our today's first speaker, Mr. Ravi Jayapal, who is a co-founder, president, and CEO of CitraGen Pharmaceuticals, California, USA. I welcome Dr. S. Amudalakshmi to take over the chat.
A warm good morning to one and all. Myself, Dr. Amuthalakshmi, Associate Professor, Baid Metha College of Pharmacy, Chennai. I am pleased to introduce the speaker of today's first session, Mr. Ravi Jayapal, in the webinar series 8 titled Advancing Scientific Knowledge in Times of Pandemic. He is presently functioning as a co-founder, president, and CEO in CitraGen Pharmaceuticals, California, United States. He has over 20 years of versatile experience in research, commercial manufacturing, and management in pharmaceutical industries. He has a sound experience and knowledge with the regulatory requirements of US FDA and other international regulatory agencies, and he had been a part of regulatory filings, approvals with US FDA for more than 100 drug products. Mr. Ravi sir has a strong knowledge and hands-on experience with analytical techniques and instrumentation involved in pharmaceutical research, regulatory, quality control, and manufacturing. He is a research expertise and he has headed various international and national pharmaceutical companies. During 2000 to 2004, he was ahead in analytical R&D in Aurobindo Pharma Limited, India. During 2004 to 8, he served in Concord Laboratories in New Jersey. In the middle of 2008 to 13, He was a proactive director in AR and QC in Pharmadex, which is a pharmaceutical contract development and manufacturing organization in Stockton, United States. To his credit, he has filed about 90 abbreviated new drug applications with US FDA in 4 years. He has created internal systems and managed small to large research groups and handled a wide range of products such as OTC, ANDA, IND, and NDA. And he has also played many leading roles in the administration, research, regulatory, business, and marketing. Sir has also played many key roles in business transformations and development of pharmaceutical drug companies. In today's session, Char will describe the impurity identification, characterization, qualification, and analysis in drug products, and overview on industry practice. This is a very interesting and trendy title, especially for our students, sir. Sir, once again, welcome you, sir. The session is over to you.
Thank you. Thank you, Lakshmi. Thanks for a very nice introduction about me.
So a warm welcome to Mr. Ravi Jayapal, sir. Hello, sir. Good morning.
Good morning, Vijay. Good morning.
Good morning.
So now the session is over to you, sir. You can start the session.
Thank you. Good morning to everyone. I hope everybody is staying safe and staying at home and in the very different pandemic situation. We are all getting used to the new normal. Hopefully everybody is staying safe with the family and friends. I request you all to stay home to help the pharma industry, to concentrate on the new drug developments to help the pandemic to get over. So in today's session, we're going to talk about the impurity identification, characterization, qualification, and analysis in drug products. Before going into the topic, I would like to thank to everyone in the Baid Metha College of Pharmacy for giving me an opportunity to talk to you all. I hope this will be useful and knowledge gaining for everyone. So moving on to the topic. So this, since it is a very big topic, I cannot cover each and everything in this one session. So probably what I tried to give is an overview of overall impurities, this one. So I splitted this into two sections. So one is I will give a background about what are impurities and what are the types of impurities and what are the major current global requirements for the impurities in the drug products specifically. So since when we cover the drug product, we can cover excipients, API, and everything. And I will give you an overall practice or overview of the practice, how an impurity profiling is done on the drug products. So maybe I'll give you an example how we identify, how we characterize, how we qualify, and how we analyze these impurities. So moving on to the topic. So before going into this one, first we all should understand what are impurities. So by definition, any component of a drug product. So drug product means any tablet or capsule or any liquid oral or any of the drug product which we commonly use as a dosage form, which we consume as a patient. So any of the drug product or any component of the drug product, which is not a drug substance or an excipient, which we added into the drug product. So what does this mean? So say for example, take an aspirin tablet. So this is the formula which we manufacture aspirin tablet. So this aspirin tablet, it contains aspirin, which is an API, and then it contains various excipients, microcrystalline cellulose, mannitol, crospovidone, magnesium stearate, sucralose, then colors, flavors. So only these are the components which we added to the drug product. So anything other than these components are impurities. So where are all these impurities can come from? Any component that is degraded from your API, any component that is a reaction, residue, or byproduct from the raw material, which is any API or excipient. Since we synthesize all this API and excipients, there can be reaction residues, there can be reaction byproducts. So those all are considered as impurities. Even though we purify them and we remove them, we may not be able to remove 100%. So there will be some trace amount of those residues will be there. So those has to be characterized and qualified. Any component that is degraded due to interaction between the API and excipient. Say, for example, we added aspirin. So that can react with any of these excipients. So we don't know that. So if anything is reacting like that, and if it is forming any impurities, so that is also called as impurities. Then we pack this dosage form into the bottle or blister pack or anything. So any component, if anything is reacting with the packaging component, and if it is forming anything, or if anything comes from the packaging component to this one, so that will be also considered as impurities. Similarly, any component that is degraded during the manufacturing process of the drug product, say for example, we manufacture this, we do granulation, we do compression, we do coating. There are so many unit operations during the manufacturing process. So during that process, the drug and the excipients are being exposed to extreme manufacturing conditions. We might be heating during the coating, we might be drying the material in a oven at 60 degrees. So these are all extreme conditions. So those times the drug product can form any impurities, either degrading from the excipient or the API. So that is also as impurities. Also, any component that is degraded during the storage. So that's what we call it as stability of the drug product. So that is also impurities. So now, so many of these are there. So what are the common components, or how do we identify which are impurities? So for that, the overall thing is we classify them into major categories based on the chemistry. So when we talk about chemistry, it is 4 major categories we usually do it in the industry. So one is organic impurities, and then inorganic impurities. Then there is one more sub topic from the organic, which is volatile organic impurities. And then moisture. Yes, moisture is also an impurity because we don't add water into the formulation. Say, for example, in a tablet or capsule dosage form, we don't add water. We might be using it for granulation or coating, but we are removing during the process. So that any amount of trace amount of water is there, that is also considered as an impurity. Also, any water can permeate during the storage, or this one, say for example, in Chennai, if we take, it's very high humid condition. So if you put any tablet in a HDPE bottle and keep it, it can permeate the moisture from the environment to the tablets. So that is a stability issue when you are storing the product. So those also considered as impurities. So when we look at the organic impurities, we classify them majorly into 3 different categories. So one is degradation products. So that is any organic compounds degraded during the storage. Usually it happens in the API. Say for example, in the aspirin case, aspirin degrades into salicylic acid, which is very well known degradation pathway. So same way, organic compounds degraded during the manufacturing process. We use the heat, we use water during the coating or anything during the manufacturing operation. So those are called degradation products. And then there are process impurities. So these process impurities are formed during the manufacturing or during the reaction of synthesis by byproducts or residues from the raw materials of excipients or anything. Similarly, organic compounds can leach from your packaging materials. This is commonly happens in the liquid drug products like injections, oral liquids, ophthalmic drops, etc. Because we store them in a HDPE bottle or in a glass bottle. They all have certain amount of colorants, they have stabilizers, they have plastic components, so those can leach into your drug product. So those are leachable organic compounds or leachable impurities. Similarly, inorganic impurities. So inorganic impurities are the one which is metal residues. Say for example, we do a synthesis, we might be using a catalyst. So that might be an inorganic catalyst. So from there it can come from the impurities. Also, the process equipments are made up of stainless steel or aluminum, or mostly it is different metals. You have transfer pipes, which is made up of silicone, or which is made up of metals. So these all can come from organic impurities like inorganic impurities like lead, arsenic, mercury. Like those heavy metals can leach from these components. Also, the inorganic components can leach from the packaging material. Say, for example, we have aluminum containers, so aluminum can leach from there. So also, a lot of plastic containers have a lot of rubber caps, and all these ones have the inorganic salts or inorganic metals as stabilizers. So those also can leach from the containers and the closures to your drug product. So that is all classified as together as inorganic impurities. And then the volatile organic impurities. So the volatile organic impurities are a little bit narrow class. So this is basically mostly the residual solvents which we use in the API and the excipients processing. So they are made during the drug product manufacturing process or during the purification of the API or the excipient. So those ones are the volatile organic impurities. Similarly, volatile compounds can leach from the primary packaging components, since those also have certain amount of solvents and all those stuffs. So these are the major or broader category of the impurities. So now we have to look at what are the acceptable limits before going into the characterization. And this one, we should know which we should identify and how to identify. So for that, we need to know what is the acceptable limit, because we cannot keep going. You can find 0.1 microgram with your method, then it might be more sensitive. You need 0.1 nanogram. So you have to keep going lower. So there should be some basic standard where we should start. Okay, this is the minimum standard. So from above this, you have to do it. So below this, how you're doing it is individual case-to-case basis on the product. So for this purpose, a global effort has been generated for years together, and then they formed a committee called International Conference of Harmonization. So this committee, what they give is they recommend different limits for different type of impurities. So they have made different guidelines for different class of impurities. So for organic impurities, the guideline, ICH guideline, which broadly describe detail about the recommendations, is ICH Guideline Q3b, which is currently revision 2 is in the effective version. So it takes a risk-based screening approach based on the daily dose of the drug product. So the approach is here is first report, then identify, then qualify. So for each of these levels, they have created a threshold-based limits on the basis of maximum daily dose and the total daily intake. The total daily intake of the impurity and the maximum daily dose of the drug product. So the reporting threshold, any impurity above this threshold must be reported. Commonly, these are called as unknown impurities. And identification threshold. So any unknown impurity, when it crosses the identification threshold, it should be identified. So it should be identified means its structure has to be established, or its source of impurity, where from it is degrading or how it is coming, that has to be identified. Then the third one is qualification threshold. So the qualification threshold is when the identified impurity is crossing this limit, the impurity should be qualified based on their toxicity if the limit is crossing the qualification threshold limits. So for this purpose, they have classified the doses with this way. So the doses are, for reporting threshold, the doses are classified in two major categories. So one is less than or equal to 1 gram. So any drug product which is less than or equal to 1 gram, any impurity up to 0.1% or any impurity less than 0.1%, you don't need to report it. It can be called as unknown impurity below threshold limits. Say, for example, it is your less than your deduction limit or less than your quantification limit. So this is for less than 1 gram dose. So up to 0.1%, you don't need to report that impurity. If the dose is more than 1 gram, then any impurity above 0.05% should be reported in the reports on the chromatogram or in your certificate of analysis or anywhere. It should be integrated and it should be reported. So when we come to the identification threshold, the dose becomes more narrower because now the unknown impurity becomes a known impurity. So now you have to identify the structure of this impurity. So that means more tighter dose limits coming into the picture. So when it is less than 1 mg, you can, if any impurity is more than 0.1%, up to 1%, you can keep that impurity as an unknown impurity. And then if it is 1 mg to 10 mg, then it is 0.5% or 20 microgram total daily intake. Here the total daily intake comes into the picture because a 10 mg tablet, you may take 4 tablets a day or only 1 tablet a day. So depending on that, you have to calculate the total drug intake or the percentage, whichever is the lower one, so that you should adopt it. So that's how the threshold levels work. The numbers are a little bit confusing, so better you can see a representation in the chromatogram. Say for example, this is a analysis you did, and this is your API, and these are the impurities you have find in your HPLC chromatogram. So now you are looking at the area percentage. So your API is 99.55% pure, but only 0.45% is impurities. So now these impurities, you have to now fit into the threshold limits. Now, if you see this impurity at 6.609 minutes, it is above 0.23%. So that means it is more than the identification threshold. So you have to identify the structure and characterize this impurity. So now, if you see these impurities at 2.7, 10.6, and 12.1, those are above 0.05%. So if your dose is more than 1 gram dose, they are more than 0.05% is your reporting threshold. So these impurities you have to report in your chromatograms and in your reports. If you see the 17.484 impurity, which is only 0.02%. So that is less than the reporting threshold. So you can disregard this as an impurity. So now, in a simple terms, reporting means any report it as an impurity and not necessary to identify the structure. So that means for this 0.05, 0.06, and 0.09% impurities, you just report them as an impurity. You say it is an unknown impurity at so-and-so RRT, or so-and-so retention time. So that becomes a simple report. So now when the identification threshold, when you cross, then you have to identify what is that impurity, and then you have to tell, okay, this is the structure. So I have seen that 6.609, 0.23%. So this impurity structure is this. This is the chemical name of this. So that one we have to identify and we have to say that. So the qualification is you have to establish the toxicity based limit. So now you cross your qualification threshold. So that means that impurity has to be fully characterized. Fully characterized means you have to do a full toxicity study like acute toxicity studies, chronic toxicity studies, genotoxicity studies. All those toxicity studies we have to do and then establish a toxicity based limit for that particular impurity. So that's the threshold for the impurities. So now going on to the inorganic impurities. So most of these inorganic impurities, like the ICH Q3b is the guideline which talks about or recommends the limits for these impurities. The reason why these are come into the picture is most of these inorganic impurities, for example, arsenic, cadmium, lead, all these heavy metals, they pose a very high toxicological risk to patient without providing any therapeutic benefit. So these are commonly known as elemental impurities or heavy metals. So the consumption of these may result in unwanted health risks for the patient, even at the trace levels. Now the problem is if we go into the table of elements, there are multiple elements. So how do we monitor all these elements? So it is impossible to monitor all the elements in every drug product. So for this purpose, what they did is they have created a toxicity-based limit and selected what are the likelihood occurrence of those elements in the drug product. So this is a big exercise done by ICH in coordination with USP, JP, European Pharmacopoeia, and all these people. So they come together and they did this, and then they established this PDE limits. So this PDE limits are how they did is they did lot of animal studies, and then from there they have selected no observed effect levels and are the lowest observed effect levels. And then from that they have selected a model of animal which can represent human nature. So based on that, PDEs, 24 elements they have identified. So those 24 elements are the specified with their toxicity limits and defined as maximum permitted daily exposure levels in a microgram per day for all the 4 major drug delivery categories. So further, what they did is these 24 impurities, they classified into 3 major categories. And based on their toxicity and possibility or likelihood probability to present in the pharmaceutical, they have put together limits. So apart from these 24 elements, any manufacturer can use any other element or any other metal, but when they use it, they have to establish the limits on their own, basically what metal they are using according to the toxicity, and then they have to do that. So the classification, how they classified these 24 elements, are they classified into 3 classes. So one is class 1. So in the class 1, there are 4 elements. So these elements are non-human toxicants. That means they have limited or no use in the manufacture of pharmaceuticals, but their presence in the drug products typically comes from the commonly used materials. For example, the class 1 metals like arsenic, cadmium, mercury, and the lead. These are very common impurities even in the water. So they can come from water, they can come from your container, they can come from your excipient, they can come from your API. So these excipients, mostly they come from like magnesium stearate like products where when you manufacture from the mined excipients or the mined sources. So these can be present. Then the class 2 elements are generally considered as route-dependent toxicants. So that means if you take oral, it might be not more toxic, but if you take it in injection or in a nasal route, it might be more toxic. So accordingly, they have, based on that, they have classified this into 2A and 2B, 2 different routes. So the 2A is these 3 elements, they put it to cobalt, nickel, and vanadium. So these 3 elements are having relatively high probability of occurrence in any drug product or anything, because cobalt, nickel, and vanadium are the common alloy components in the stainless steel. Since most of the pharma equipments are made up of stainless steel, from there these 3 elements can leach into your drug product. The class 2B is 10 elements they have identified. Most of these elements are co-isolated materials. So co-isolated materials means most of these elements are used as catalysts or filters or those kind of process-based or manufacturing-based possibilities to come into the drug product. So that is the reason they have been classified as class 2B. So the class 3 these are 7 elements that have relatively low toxicity by the oral route, but they have high toxicity when administered in a nasal route or in a parenteral route. So for that purpose, they have added this. So then this, depending on your route of administration, you have to see if your PD is above 500 microgram per day, you don't need to do the risk assessment for this impurities. So in overall, the limit of these impurities are put together a table. So try to give an overall view. So if we see if one or more element of this 24 element is intentionally added in a drug product or in a drug substance, so that respective element should be controlled and evaluated, and we have to do the irrespective of route of administration, no matter what. So if it is intentionally added, you have to do it. Then class 1 and class 2A elements, whether or not you intentionally add it, it must be evaluated and assessed for the risk, because these are highly toxicants. So that has to be, no matter what which route you are doing, so these has to be evaluated for the risk. And class 2B and class 3 elements, So these are, depending on the route, the specific elements must be evaluated for risk. For example, if you see the lithium, antimony, and the copper, cupri, the copper, so these three are should be evaluated when you do it in the parenteral route. But if you are doing in oral route, you don't need to do it. Whereas for the injections, your lithium, antimony, barium, everything must be, these all marked as S1 should be evaluated, because these are very highly toxic when you give it in the parenteral route. So how do we assess the risk for the metals in a pharmaceutical product? So this has to be done in a systemic process. So the risk assessment is a systemic process to evaluate the possibility of risk. So that elements, how that element can be introduced in a drug product and exceed the PDE levels. So we have to see the possibility of risk. So typically this is done during the R&D stages of the drug product. So a typical risk assessment involves three major steps. So in the first step, we identify the known and potential sources of elemental impurities that may find the way into the drug product. So the excipient can give the elemental impurity to the drug product. Similarly, API and the drug substances can come into the drug product. So these all, we have to do the risk assessment. So the API, drug substances, and excipients are the most common sources to majority of the drug products, and it can be controlled by the vendor qualification and selection process to keep tighter control. So when we buy the API or the excipient from the vendors, we have to insist them to do the analysis and get the reports from them and we have to see their manufacturing process and understand where their product can have the impurities. So these facility manufacturing and packaging is under your control. So you are doing it. So these in your facility, you can do a common risk assessment and keep it evaluate based on product to product. The step 2 is you evaluate the presence of particular elemental impurity in the drug product, either by determining the observed or predicted level and compare it with the PDE limits given in the ICH guidelines. The step 3 is identify if controls are built into the process are sufficient to or identify additional controls. Say, for example, in the R&D stage, we find a manufacturing equipment is giving, after, say, for example, after coating, I'm getting nickel more. So that means during my coating pan is leaching the nickel into the drug product, or probably the coating excipient has it. So now you identify the problem where it comes from. Is it from the equipment or from the excipient? Then control it by controlling that particular source. So summarize all this document, that becomes the risk assessment. So the volatile organic impurities. So the volatile organic impurities is the, other than the common solvents which we are using in the manufacturing. So that goes under the same Q3b R2 guidelines, which is the organic impurities. So for the residual solvents, they have put together a separate guideline because these are a little bit different from those volatile organic components, because these solvents are more toxic than those ones. So they put together a different guideline for these ones. So that guideline is ICH Q3c. This is one of the most updated guideline or most often revised guideline, because more studies they keep on doing on the solvents, and then the limits keep revised every time. So this is one of the constantly updated guideline. So the reason is the residual solvents have no therapeutic benefit, and all residual solvents must be removed from the drug product or the API or excipient to the maximum possible. So we have to make whatever the maximum efforts to get whatever the maximum extent possible, we have to remove those solvents. So the drug products also should not contain any higher residual solvents. So that has to be supported by the safety data. So say, for example, if your drug product has to contain ethanol more than the limit, then that has to be supported by the safety data of the ethanol. So that is how. So you have to prove your drug product requires that much amount of ethanol, and that is safe to the population where you are administering. Say, for example, I cannot put 20% ethanol in a drug product and give it to a pediatric patient. So that is the one basically designs. So your patient population, your dosage form need, and your safety, all these three has to be harmonized together. So the residual solvents are classified into three classes based on their possible risk to human health. So the class 1 solvents are highly toxic, or mostly they are known as human carcinogens and environmental hazards. The class 2 solvents are the solvents, they should be limited. They are non-genotoxic carcinogens or possible irreversible toxicity such as neurotoxicity or teratogenicity or like that. So these solvents are commonly used, but they have to be limited, not like, say for example, you have a solvent which has to be needed to purify your API to remove all the impurities, then you can use that, but the amount of solvent should be limited. The class 3 solvents are the solvents with low toxic potential, but they are still low toxic potential, but their health-based exposure limit is needed. For example, ethanol. So the class 3 solvents have, usually their permitted daily exposure levels are a little bit higher. So they are 50 mg or more per day, but still they have to be limited. So for each of these class ICH put together a limit. So for the class 1 solvent, the benzene is the most stringent solvent, which we have to keep it very tighter limit. So that is 2 ppm. 2 ppm. So that is the reason it is a carcinogen. That is the reason it is very highly controlled. And then carbon tetrachloride, that is also a toxic and environmental hazard impurity. So that is also has to be controlled. Then 1,2-dichloroethane and 1,1-dichloroethane are the toxic impurities, which has to be limited at 5 ppm and 8 ppm. The trichloroethane impurity is a little bit higher limit because it is an environmental hazard. So when you use it, it has to be justified and within that limits. So these class 2 solvents are the common solvents which we see: acetonitrile, chloroform, dichloromethane, dichloroethane, like those solvents. So individual solvents have 2 limits. So if we see the permitted daily exposure, which is milligram per day, and limit. So the both, whichever is the lower one, that we have to adapt it when we take the drug product. So the class 3 solvents, they should be limited by the GMP or other quality-based requirements. The general limit is 5,000 ppm or 0.5%, but most of the times they are controlled less than this based on the actual historical data or what are the practical possible data. So how these impurities are commonly analyzed or monitored? So most of the cases, the organic impurities are identified and characterized during the product development as part of method development and using validated methods like HPLC methods. Mostly we use HPLC methods. And then every batch is analyzed as part of quality control. And then also they are monitored during the stability studies. The inorganic impurities, mostly it is done a detailed risk assessment along with the analysis, mostly ICP-OES or ICP-MS or X-ray fluorescence or other alternative techniques. And the data during the product development and process validation, which is basically your risk assessment data. So based on that outcome, these impurities are either controlled at the raw material level and or otherwise in the in-process controls. In cases where required, we have to do each batch analysis. They are analyzed using validated methods as part of quality control if potential leachable case is suspected. So most of the injectables and liquid oral products are the ophthalmic drops. They are monitored during the stability also for the inorganic impurities. In many cases, oral products like tablets or capsules, they are done as a part of process validation exercise, and then periodically they monitor it only during the change of the equipment or something like that. The volatile organic impurities, the residual solvents, they are mostly used in the API and excipients. They are controlled at raw material levels. If required, the drug product is analyzed on case-to-case basis levels. So it is depending on the situation. If solvent is used in the drug product manufacturing, because sometimes we do non-aqueous granulation or non-aqueous coating, so that is based on the class of solvents, we have to use the appropriate methods, mostly GC or GCMS. And the leachable volatile organic compounds from primary packaging materials, they are monitored as part of stability when we suspect that it is there. And moisture, the water content, it is mostly done monitored during the in-process quality control and the stability. That is done by either KF titration or the loss on drying. So now let us go into the impurity profilings. We got an overview of what are the impurities and how do we analyze and what are their limits. So now moving on to the impurity profiling. So we should understand first what is the impurity profiling. Impurity profiling, don't confuse yourself with a big high-fi term. In a simple term, impurity profiling is a list of impurities or the description of identified and unidentified impurities which is present in your drug product. So how this profiling is done. So let us take an example of two products and see step by step. So say for example, we have aspirin chewable tablet which is manufactured by a direct compression process, and then we are using the maximum doses, only one tablet a day. This is used for the prevention of heart attack and stroke. So this contains aspirin as a API, and then it contains MCC, mannitol, and all these excipients and this one. So the same aspirin, we take another product, which is delayed release tablets, 325 mg, which is used as an analgesic or antipyretic, the popular non-steroidal anti-inflammatory drug. So this one is maximum daily doses. You can take up to 48 tablets in 24 hours based on the toxicity stability data. So that means 15.6 grams of aspirin. So in this one, since it is a delayed release, we have entry coating polymer, polyethylene glycol, color, and all the coating process, everything is involved. And also it is a wet granulation process. So now let us see how we can do the impurity profiling for these two products. So where to start? The first step, always we have to start from the API. So the first step is we have to gather the impurity details from the API manufacturer or from the synthetic route. Most of the cases we purchase the API from a third-party manufacturer or we manufacture on our own. So when you purchase the API from the third-party manufacturer, always they give you a full impurity profile as per their synthesis route, because they also have a guideline to be followed, ICH Q3a and Q6a guidelines they follow. Based on that, they give you a full impurity profile. If the API is synthesized by our own, then we have to generate the impurity profile data based on those two guidelines. So the typical API impurity profile is listing all the impurities, what are the impurities possible in the API, and their source indicating whether they are starting material or reaction byproduct or degradation product or catalyst or reagent, like that. So in this case, we are looking at aspirin as our API. So aspirin is a very simple synthetic route that is just made in a single step synthesis process by acetylation of salicylic acid with acetic anhydride. So it forms acetyl salicylic acid and the acetic acid. So now let us see what is the impurity profile we have for this. So this is the impurity profile, typical impurity profile look like. So they have the impurities listed and their chemical structures. And if any pharmacopoeia or anything is there, the pharmacopoeia impurity names, and then the source of impurity, and then the type of impurity. So say, for example, here the 4-hydroxybenzoic acid, it is trace impurity from the raw material. So that means this is an impurity coming from the salicylic acid raw material which they are using it. And this is a type is organic impurity. And then if we see the 4-hydroxyisothalic acid, so this is also, it is trace impurity from the raw material. So that is also from the raw material of the API. So this is also organic impurity. So now they have the impurity C, which is salicylic acid or 2-hydroxybenzoic acid. So which is a process impurity because it is the starting material, and also a degradation impurity. We all know that aspirin can degrade into salicylic acid. So that is a degradation impurity. So this is also an organic impurity. So similarly, they put impurity D, which is a reaction byproduct, and then impurity E and F, which is process impurity, and then again reaction byproducts. So they have. So if we see here, they have put a star mark on 4 of these impurities, and they say always below 0.03%. So ICH Q3 reporting threshold limit for them is 0.03%. So it is always below that limit. So that means these impurities are possible to present, but it may or may not be present in the API which you get it. So that means since it is less than 0.03%, dose-based ICH evaluation we have to do. So for 75 mg, 0.03% is less than the 0.1% threshold, because 75 mg tablet we are taking only 1 tablet. So that is falls under the less than 1 gram dose. So the less than 1 gram dose, the threshold which we have seen in the ICH is 0.1%. So this is less than the identification threshold. So that means we don't reporting threshold. Similarly, if we look at the 325 mg, the 325 mg is less than 0.05% because it is more than 1 gram dose. So the 0.03% is still less than 0.05%. So that means these 4 impurities you can monitor, but you don't need to report it. If it is growing above your limits, then you can report it. Whereas if you see the other 2 impurities, the salicylic acid and acetyl salicylic acid, those 2, they didn't say anything about the levels. So since they are typically more than 0.03%, and also salicylic acid is a degradation impurity, so this must be included in the drug product monitoring as a known impurity. So similarly, now they have given a ICH class 3 solvent, which is acetic acid. So acetic acid is their source of impurity. They are saying it is a process solvent and degradation impurity. So it is basically, it is coming from the process byproduct. When they synthesis, they have acetic acid in the reaction. So even though they remove it, it might be still in the trace level. So they're calling it as a process solvent. Also, when your aspirin degrades into salicylic acid, you get this acetic acid also as a degradation product. So this is this one. So that means since it is a degradation, this also we have to include in the drug product impurity monitoring. So now the next is inorganic elements. So they are saying they have used no reagents or catalysts, ligands that are potential to add inorganic elements, including heavy metals. So that means they haven't used any of the catalyst or anything. So on all equipment, they are saying it is made up of stainless steel. So the process validation type and stability studies and periodic QC analysis of their results show no potential of ICH class 1, class 2A and class 2B elements to be present. So that means they have did their process validation, they have did their stability studies, and they say from none of this one, there is no potential for these impurities. So that means since there is no potential for these impurities, we have to do the risk assessment based on the other components like excipients and packaging components. So we don't need to right away decide on this. So next is the inorganic salts. So the elements and salts, the differences is the elements are, they are talking about the pure elements like lead, arsenic, cadmium, and those ones. The salts are the salts which they might have been having in like a sodium sulfate or other ones. So here they are telling the sodium sulfate salt is there in the salicylic acid up to 0.04% of sodium sulfate. So that means this one can be there in the traces of product. So this they are removing it during the process, and they are doing sulfated ash as a part of routine QC test, and that is typically 0.06%. So that means it is QC controlled trace salt of in API. So we don't need to list this as an element since we going to control it in the quality control of the raw material. So now let us look into the excipients. So the excipients in general, if we see the organic impurities from the excipients, they are considered as low risk for 2 reasons. One is most of the excipients are insoluble or non-digestible or non-bioavailable to the human body. For example, microcrystalline cellulose, HPMC, and all these. These are cellulose polymers which our human digestive system cannot digest this. So there is no possibility of any impurity or any degradation products are there. And moreover, when these excipients are soluble also, these organic impurities are well characterized with the known toxicity profiles. For example, mannitol, if you take the mannitol impurities, sorbitol, isosorbitol, and all those ones are again used as excipients. They all have very well characterized toxicity profiles. So most of them don't degrade further on the storage. So that's the reason they consider as a low risk. So to help the industry, various regulatory agencies like FDA, UK, MHRA, and they all created a centralized database system where they say, okay, up to this level, when you use this excipient, these excipients are safe and sound. So you don't need to worry about the impurity profiling of these excipients. So those ones, they have given different limits for those different dosage forms. So those ones, we can take that as a guideline and we can use that. So when these excipients are used within those guidelines, they are considered as safe excipient, safe level. So we don't need to do any further this ones. But the other part is we have to prove the interaction between the API and the excipients. So this is done by 2-level studies. So one is during the drug product development, a drug excipient interaction study is developed to risk assess the suitability of individual excipients. You will do like every individual excipients, aspirin plus MCC, aspirin plus mannitol, aspirin plus this one. You put it in this one and you conduct a small stability study, basically like 1 month, 3 months, like that. So this is called compatibility studies. So that gives you an idea about how these excipient can interact with the API or how that can degrade individual. So based on that, we can select the excipients and that we can use it in our formulation. And further, what we are doing is during the stability studies, which is for your product, you have to do the stability studies to establish the shelf life of the product. So expiry date, which we put it on the label. So during that stability studies, we monitor these impurity levels and then we further ensure that there is no interaction between the API and the excipient. Other inorganic impurities and residual solvents used in the manufacturing process of excipients are controlled during the QC of the excipients. And we have to further do the risk assessment in the drug products as per the ICH guidelines. So how we do that? So we have to list the excipient. So what are the listed residual solvents and what are the listed inorganic elemental impurities? This information we can get from the individual excipient manufacturer or supplier. So in my MCC, they haven't used any of the solvents and they haven't used any of the elements. In mannitol, they haven't used any of the solvent, but they have used nickel as a catalyst. And they say it's typically less than 0.5 ppm and the limit is, for them, the quality control limit is 1 ppm. Similarly, for the povidone, they use acetone as a solvent, but the limit is less than 200 ppm, usually the typical results. So they give the typical results based on their historical data of analysis. So usually it is less than 200 ppm, but the limit is 500 ppm. So they don't use any of the metals. Crospovidone, they use ethyl acetate as a solvent. It is typically present less than 150 ppm and 250 ppm. So like this, we have to collect all the information of individual excipients. So once we have all this data, so now we have to get into the impurity profiling, how we do it. So during the product development, we have to do the drug excipient interaction study to select and identify the excipient that will not interact or degrade the drug. And then we have to do a force degradation study on the drug product to understand and to identify the possible degradants from the API and our excipients. Because when you subject it to acid, alkali, oxidation, temperature, and humidity extreme conditions, if any of the interaction is possible between the API and the excipient, you can find those impurities on these force degradation studies. So in this force degradation studies, we do the tablet, if you are doing, the whole tablet we take, and then the whole tablet powder we do the force degradation, not just the API force degradation. So that way you get that full information. Similarly, in both the above cases, you monitor the known impurities. So you have already identified what are the impurities, the impurities from the API, the list, and then based on the data of that, you monitor any unknown impurities. So that could possibly come from the your API excipient interaction, or your drug product is making your API to degrade more than your API. So those can also be possible. So now you have to see the unknown impurities, and if required, you have to use the LCMS or NMR to characterize and find the impurity structure. If they are crossing the ICH reporting threshold, then we have to go to the toxicity-based data, and everything we have to do it. So based on these informations, you create a tentative impurity profile of the organic impurities based on the API impurity profile, because API has already given you the data, what are the process impurity, what are the starting material, what are the byproduct, so everything you have from them. So now based on that, you add your information which you generated during your force degradation studies and excipient, your interaction studies, and then create a tentative profile. Based on that profile, you validate that analytical method, and that you use it for your stability monitoring. So now you have the base method. So now the manufacturing or the product development, they are going into the process development and the scale up. So now in this case, you identify the critical steps where the monitoring of these impurities using the above validated method you require. Say for example, they are doing coating. So monitor the impurities. So during the coating, any degradation is happening or not. So before coating, how much impurities? After coating, how much impurities? So those levels we do that, and then based on that, say for example, your drug is degrading during the coating process. So that means it can cross the identification threshold or qualification threshold. So that means you may have to be prepared for creating your toxicity studies or initiating toxicity studies to go beyond those limits, which is there given in the guideline. Then if the solvents are used in the process, monitor the removal process. Like say for example, you do wet granulation, see the drying process, ensure that your moisture content is going down, or ensure that your residual solvent is going down up to within the ICH recommended guidelines. So say for example, you use acetone in your coating process, dry the tablet, see whether it is below 5,000 ppm. If it is below 5,000 ppm, dry it still further to see whether your impurity is increasing or your acetone is going down. So you have to make sure balancing the process. So that is where the whole analytical support is needed in the product development as well as in the manufacturing. So then evaluate the potential for the inorganic elements that can leach from the processing equipments, or create a risk assessment for data for the elemental impurities. So now the risk assessment for the elemental impurities. So now we have the individual drug product, API and excipients. We know from what are the excipients has this one. So from the API, they haven't added any intentionally, they haven't added any of the catalyst or anything. So except in this product mannitol we are having. So mannitol, they are using nickel as a catalyst. So that is the only one which is intentionally added. So potential elemental impurities. So this class 1 impurity, since it is a highly toxicants, we are considering this as potential elemental impurities to be present in all the excipients. Because even if it is there in one, then it is a problem. So we consider this on the risk assessment, it is potential. So potential elemental impurities that can come from the manufacturing equipment, based on the process we have to see, okay, we are doing the granulation in the, say for example, we are doing the granulation in the high shear granulator. So that is made up of stainless steel. So you can get vanadium or nickel or cobalt or so those ones. So that is from the manufacturing of the equipment. So then process potential elemental impurities from the container closure system. In this case, it is, since it is a tablet product, in a solid state, you don't have a possibility of leaching from one solid to another solid. So that means that becomes minimum risk here. So that becomes none. So like this, we have to do for both the cases. So now when we do it, so then the risk-assessed element as per the guideline, we have to, whether we add it or we don't add it, we have to do the class 1 and class 2A. So now we don't have any other element has been added. Say, for example, your API manufacturer used palladium as a catalyst, which is a common catalyst. So then we have to do the palladium also, we have to add it to the risk assessment. If not, then just only these 7. So now, how do we establish? So we have 3 options. One is drug product analysis option. The other one is summation option. The third one is individual component option. So let me explain that a little bit better. So the drug product analysis option is we just take the drug product, we analyze the drug product by ICP-MS or any analytical method, and then you scale that whatever the impurities elemental impurities you found into the maximum daily dose or into the PDE value. So that is calculated by, so whatever the measured value in your microgram per gram and then maximum daily dose in gram per day, you end up with microgram per day. So for example, this product we analyzed. So this one we got 0.11, so these are the limits. So the maximum daily doses, the 325 mg is the higher dose. So that has been taken as a worst case. So then that with the maximum daily dose, when you multiply, so it comes here. So here the difference is the maximum daily dose comes to the total tablet weight, not the API dose, because here your elemental impurities are included from the excipient. So you have to consider the total tablet weight of as a dose. Otherwise, you will be missing up those calculations. So that should be considered. So with that, you have to calculate what is the PDE you got. So using this formula. So we multiply this 2 and we get this PDE, microgram per day. So the limit is this one and the amount what we got is this one. So the result is passing. So now that means you created a data during your process validation by analyzing multiple batches of your process. So with the set defined manufacturing conditions, you establish a data saying that, okay, my product, even though it might contain some elemental impurities, it may not cross the limit. So this is the risk assessment process, and then you freeze this here. So whenever you change your process equipment or whenever you change your manufacturing process, only at that time you will do it. Otherwise, you will not do it. But in this case, will change if you have added any impurities or any catalyst to the process. And if that is there in the part of that, then every batch you might have to do it. Or if your equipment, say for example, your equipment is leaching, then your input and the output is different, then you have to do it. The second option, the summation option is it is another option which it is usually does. So you take individual elemental impurity microgram per gram present in each of the components, and then use your drug product formula, what the ratios you have of each of them, and then accordingly you sum it up that, and then the total together, you see whether you have this option, whether it can be do it. So this can be used as a substitute option, or you can use this when you have a documented evidence that there is no additional elemental impurities can be added or can be come from the manufacturing process or via the container process system. So only at that time you can take this option. The individual component option is, this is a unique option. This is only given, recommended by only the USP. So when your dose is less than 10 gram per day, daily dose, if all the drug substances and excipients meets a certain limit, which is less than the ICH or USP limits, so then you can use that without doing any analysis further in your drug product, because that will fall under this one. So you can see that limits, individual limits there in the USP general chapter, so you can see there. Then the risk assessment, how we do for the residual solvents. So the residual solvents, so the drug product manufacturing process. So in this case, we are not using any of the solvents. So that means we are less from there. If we are using any solvent, then we have to analyze that solvent. So the drug substance, they haven't used any solvent, but acetic acid is the degradation product which comes from the API. So also it is possible to degrade in the drug product, and it can come from the during the post degradation studies. Also, we can see the acetic acid levels go up. So that becomes your drug excipient interaction study results. Because of that, you need to monitor that ICH class 3 solvent. So now that has to be monitored. So in the excipients, some excipients, they have used the ICH class 3 solvent and class 2 solvent, but they are way below than the ICH limits. And these solvents cannot come during your stability or anything due to degradation, because these solvents are added to purify or added to like a process impurity. So that means they cannot grow in your stability. So that means these solvents are low risk solvents. So you don't need to monitor this. So therefore, you can make a GC method to quantify acetic acid and validate that. That method should also monitor these solvents. So that way, if you get any problem in the quality control, say for example, the manufacturer said only 25 ppm it will be there, but you got 30 ppm, or suddenly something happened, or somebody mixed in the drug manufacturing, or any mishaps happened. So you can monitor that during your final quality control when you have it in the same method. So if your drug product doesn't have any degradation solvent, say for example, here we have the acetic acid, but if your drug product don't have any degradation solvent, you can use the summation option similar to the one which we have seen in the elemental impurities. So you don't do the analysis, but you will just sum it up, and then you will say, okay, there is no risk in the residual solvent for this drug product. So in summary, this is the drug impurity profile you get for your drug product. So you have the 4 impurities, impurity C, D, A, and B, which are trace impurities listed in the API. So 2 are the trace impurity from the raw material of the API. 1 is the process impurity and the degradation impurity. So these 2 impurities are your major organic impurities. So these 2 you are analyzing by the HPLC. So other 4 trace impurities, which is reported as less than 0.03%, they are possible minor organic impurities from the API, expected to be always below the ICH identification threshold. But for safety purpose, you are monitoring that by the HPLC method which you are using it. So you won't validate these 4 impurities in your method, but you will monitor. Say for example, you will prove only the specificity. You won't do the accuracy, recovery, and those steps, but you will just do only the specificity for these 4 impurities. But these 2 impurities, you will do the full validation. So now the residual solvent, so acetic acid, we can analyze by the GC method. So on the same GC method, we can monitor these other minor impurities, the trace ICH class 3 and class 2 solvent, which are expected to be present from the excipient, but always it will be less than the ICH threshold limits. So now the class 1 and class 2A elements, the elemental impurities, 7 elements, they are potential ICH class 1 and class 2A elements, but these elements are risk assessed and periodically evaluated by XRF or ICP-MS, because we have seen here with the risk assessment, it is not there. So you don't need to do every batch analysis. You can do the risk assessment, and then periodically you can evaluate maybe every year or every 10 batch or every 100 batch based on your own GMP process manufacturing. So always when you do that kind of risk assessment, so now if we change any of the excipient vendor or the API vendor or the processing equipment, then this impurity profile has to be redefined based on the new vendor and their specifications, and then we have to revalidate the method. So that's the whole process. So this is a simple overview on this one. I hope I tried to give you an overview of the idea. So this probably give an idea. So you can further read the ICH guidelines. So I just gave the ICH respective guidelines. So you can go through that. So I'll be happy to take any questions if it is there.
Hi, sir.
Yeah, Ish.
Yeah. So that's really a nice presentation, sir. So we have a few questions from the participants. I will just display on the screen. Please answer.
Sure. So how are the biopharmaceutical impurities are characterized? Is there any possibility of unknown impurities? The biopharmaceutical impurities are also characterized in the same way. So there are only the little bit differences. So the differences are instead of organic, you will look at the biological risk. So that is the only difference. Otherwise, it is the same. Okay. The marketed, the impurities, specific impurities manufacturing for the marketed APIs in pharma industry, I believe that there is an opportunity in synthesis, but there are many service providers here. So probably you have to establish your market research data and then you have to decide. Yeah, you can identify any impurity M by Z value is only a starting point. So M by Z value is same like your retention time in the HPLC. So M by Z is the basic information you get for that particular peak. So any impurity characterization or structural elucidation, it cannot be done by one single technique. So you have to start with the technique and then have to ensure it with the different technique.
So here is the last question, sir. We have picked up.
3D printing of tablets, no, they don't reduce or eliminate the process-related impurities because, in fact, it is increasing. So most of the 3D printing tablets are, it either converts the solid to the liquid to solid or build the crystallization techniques. So there are further possibilities of process impurities are there. Impurities are, as long as you use the API and excipient in your drug product, you will have the impurities in your drug product.
Yeah, thank you very much, sir, for such a answers for the query. Now I request Dr. S. Amudalakshmi to give the concluding remarks for the first session. Hello, Amudalakshmi. Is it audible to you?
I think she's on mute.
Even I think she's unable to hear us. It's okay. Hello, Amudalakshmi. Yeah, it's okay, sir. So I feel it's pleasure to thank Mr. Ravi Jayapal, sir, who has given such a wonderful presentation on the impurities in pharmaceutical products, and he has also given case studies related to all the products. That is really well explained, sir, because he has given examples with case studies and even in the pharmaceutical products, industry practice, all those things. So I'm very happy to have your presentation on board. I hope all the participants could have enjoyed the session. So thank you very much, sir, for your valuable time and sharing your expertise and knowledge in the impurities job.
Thank you, Isha.
Thank you. So now we have our second speaker on board, sir. We will be going for the second session. Thank you very much, sir.
Yeah, thanks. Thank you, Isha.
So now, dear participants, now it's time to go for the second session by our renowned speaker, Dr. Vijaya Ratna ma'am, who is a retired professor from AU College of Pharmaceutical Sciences, Andhra University, India. Now I request Dr. Vijaya Gita, Assistant Professor of Baid Metha College of Pharmacy, to give introductory note for our second speaker, Dr. Vijaya Ratna ma'am. And before going to that introduction for Dr. Vijaya Ratna ma'am, let me take few minutes time to have a glance look about Baid Metha College of Pharmacy. So Baid Metha College of Pharmacy, which is located in Tamil Nadu, is an ISO certified institute and which is having 40 years of academic excellence. It was established in the year 1980. So coming to some of the highlights related to the college, it is offering a number of courses like D.Pharm, B.Pharm with lateral entry also, PharmD and M.Pharm with different specializations like pharmaceutics, pharmacology, pharmaceutical chemistry, pharmaceutical analysis and PhD program. Coming to the admissions, they are open for 2021. and they can contact on the numbers who are interested. So some of the highlights related to the CL Baid Metha College of Pharmacy include it is ISO certified institute and approved by Pharmacy Council of India as well as AICTE New Delhi. It is affiliated to the Tamil Nadu Dr. MGR Medical University. It is the first self-financing pharmacy college in Tamil Nadu. It was established on 13th July 1980. There are no capitation or donation fees for this college. So let us have a look of some of the glimpses related to academic achievements of CL Baid Metha College of Pharmacy. So here we can see the pic of a student receiving gold medal for standing overall first in B Pharmacy during the academic year 2014 to 18 from Dr. MGR Medical University. Here is another student receiving gold medal for standing first in M Pharmacy of Pharmaceutical Analysis Specialization. So here is inauguration of the programs at CL Baid Metha College of Pharmacy. Here there are some students who are receiving the first and second prizes in different activities like essay competitions and some other poster presentation like that. So coming to the culturals and sports, the students at CL Baid Metha College of Pharmacy are very active, and these are some of the photographs where the dance team at 71st IPC they stood first place and they have received the prize. And here the winners of intercollegiate sports and cultural meet at Sri Ramchandra University. These are some of the photographs of the active participation of the students of CL Baid Metha College of Pharmacy. So coming to the Rotaract activities and NSS activities, these are some of the photographs related to the active participation of the students of CL Baid Metha College of Pharmacy in different social and responsible activities. So with this brief presentation, now I would like to invite Dr. G. Vijaya ma'am to please introduce our today's second speaker, Dr. J. Vijaya Ratna.
Good morning, everyone, and we welcome everyone to the session, Analysis of Variance in Research and in Design of Experiments. Myself, Dr. R. Vijay, Assistant Professor, Department of Pharmaceutical Analysis. It's my privilege to introduce our speaker, Professor J. Vijaya Ratna, is a renowned personality in the field of pharmacy, and it's a retired professor at AU College of Pharmaceutical Sciences, Andhra University. She is a subject expert in pharmaceutical technology, regulatory affairs, statistics, and pharmacy practice. Professor Vijaya Ratna started her career as assistant professor, later is promoted as professor, and she has total 38 years of experience in teaching and research, and also served as chairperson, Faculty of Pharmaceutical Sciences, chairperson, Board of Studies, and coordinator, PharmD course at Andhra University. Professor Vijaya Ratna has more than 100 publications to her credits. She has 3 patents and 5 funding research projects. She attended and acted as a resource person or chairperson for around 80 plus national and international conferences and symposiums. She has guided more than 100 M.Pharm projects and 37 PhD scholars. She has received the AP Best Teacher Award on 2018 and also ML Corona Memorial Gold Medal for standing first in B Pharmacy at All India level in 1979 and ML Corona Memorial Prize Best Paper published in the Indian Journal of Pharmaceutical Sciences by IPA. She has won silver medal in both B.B. Patel and GATE Agreement in Drug Prize in India for the National Essay Writing Competition. First prize in skill test which was conducted by the Canadian online platform Pharma Infonet. She's a life member of several professional bodies like IPA, APTI, IB, IPGA, etc. And she is also a fascinating individual, and we are grateful for her presence, ma'am. So now let's welcome Professor J. Vijaya Ratna, ma'am. We welcome you, ma'am, to describe about today's session, Analysis of Variance in Research and the Design of Experiments.
Thank you, madam.
A very good morning, madam. Welcome, madam, Dr. Vijay.
Good morning. Thank you.
So now it's time for you to start the session, ma'am. The board is yours.
Now can I start? Is it visible?
Yes, ma'am. Yes, madam. Everything is fine. You can proceed.
Good morning, all dear friends, students, colleagues, scholars, and all. A very good morning to all of you. It is my pleasure today to be on this platform. I thank the organizers for inviting me to talk on a topic which is very near to my heart, which is a statistics topic. but is very much a lead, very much a part of design of experiments, quality by design, and research and development. So dear friends, before I even get into my slides, I would like to tell you something. Before I start my lecture, I want to tell you something. Only a few days ago, WHO has made a statement. When everybody was talking about the coming out of the vaccine, WHO said, India is very strong in manufacture and can be counted upon to supply medicines to the entire world. It is really the pharmacy of the world. That's what everybody says. But it went on to say, it is only in the field of research and development that it is a little bit lagging behind. That has been there, that statement has been there in the air since long, my dear friends. And it is high time we recognize that and we work towards getting it off our board. We should not be lagging behind in anything, definitely not in research and development. Why are we backward in research and development? Okay, there may be many reasons. We will not go into all those reasons. I would like to tell you that we should specifically concentrate on all those issues which are making us lagging behind in research and development. And we should work on understanding and getting up those knowledge, those skills, those attitude, and whatever else that is required, which will make us strong in research and development. Our today's topic is something which is going to empower us in the field of research and development. So this is all about, basically then, to get into my topic then, you can say this is the part of the introduction that I am giving. So I would like to introduce the topic to you. I would like to tell you about one-way, two-way, three-way ANOVA. Then I'll go on to tell you a further application of analysis of variance, how it becomes useful in the conclusion, in summarizing and interpreting your results. Then I'll tell you how ANOVA plays a huge role. The learning of the ANOVA plays a huge role in design of experiments, which is a part of the quality by design. So then I'll spend a few more minutes on this introduction. So what is this? This is a statistical tool. ANOVA is a statistical tool. By tool, I mean it's an instrument. It's an instrument that helps in interpreting our results of the research. So how does it help in R&D? It helps in estimating the quality. It plays a role in quality by design. So let me spend a minute here in quality. So quality is the most important thing. We should all work towards having the highest level of quality in our work. In our work, by I mean in everybody, we are all doing one angle of pharmaceutical field. I am teaching, somebody is learning, somebody is doing research, somebody is working in a pharmaceutical company. In a pharmaceutical company, there are 100 different varieties of work things that are going on. Maybe research, maybe development, maybe production, maybe quality control, maybe research and development in analytical area, in so much. Till now you are hearing about a wonderful presentation on impurity profiling. So that's all a part of making better quality, working towards better quality. So in making our products of better quality, there are a lot of things to do. And one of them is to see that you select the best component that is coming from your research. And the way to select the best from your research is to do analysis of variance. So what then is analysis of variance? It is a statistical procedure designed to analyze the difference between the means of two or more than two samples. When two means are to be compared, we use the t-test. When more than two means are there, we use the analysis of variance. Well, throughout this procedure, I'll be talking you can say, in two angles. One angle is I'll be talking about the exact problem or statistics or working out calculation. That angle I'll be talking. In another angle, I'll be talking about how this is, what is the philosophy, what is the concept. So I'll be at the same time trying to explain the concept to you, calculation to you, and its relevance in the field of analysis, in the field of research and in design of experiments. So it's a statistical procedure to analyze the difference between the means of two or more than two samples. Well, in a particular research experiment, we have some samples. We have taken some samples and we are working on those samples. Let us suppose one candidate has taken three plant materials. He is testing those three plant materials for their nootropic activity, let us suppose. So from each he will take an extract and he will find out. And now he wants to know which plant is giving the material that is the best. And with that material, he wants to proceed further and develop a herbal formulation. So here he is making a comparison between three. Now, a simple rule here is that if you are comparing only two plants, it is the t-test for you. If you are comparing more than two, then you have to do the analysis of variance. Then I will tell you something, an experience which we had long back, about 12 years back. One of my research scholars was making a comparison between generic medicines and branded medicines. There was a widely held view that branded medicines are good, effective, generic medicines are not so effective. So my scholar was going and interviewing doctors and all those people, and one day she returned and told me, "Madam, this is the question I faced." What is that question? Look here. The doctor asked her, is statistics required to declare that a king is different from a beggar? That a child is different from an old man? Definitely it is not required. So what was there in the doctor's mind was that a branded medicine is so much different from a generic medicine that it is like a king in comparison to the beggar, which is the generic medicine. Now we all know, even 12 years ago we established that generic medicines were equal in effectiveness to branded medicines. It is the company that is making, it is the excipients that are there in the product that are different, but no way a generic medicine is so inferior to a branded medicine. This point we proved even 12 years ago and published it also. So now there is ample evidence. Now every state government has taken the policy of opening Janaushadhi shops where generic medicines are being sold in every shop and people are buying, and people are finding, doctors are finding that generic medicines are as good as branded medicines. It is only to be taken care of that the company which is manufacturing the medicine is following the good manufacturing practices. That's all. Are they meeting the requirement or not? So coming back to the statistics part, if really we are making a comparison between a child and an old man or a king and a beggar, we don't need any statistics. We simply can say there's a lot of difference. But in practical life, situations are different. Some difference will be there, but we cannot tell whether it is significant or not. A decision has to be taken based on whether the difference is significant or not. In real life, in practical situations, taking a pharmaceutical industry into consideration, every day the manager or supervisors or the R&D managers are faced with situations where they have to take a decision. I can give you some examples. Suppose they have to make a comparison between one type of mixer and another type of mixer for a mixing process. So both are there, both are giving out mixing. Which is better? Is the difference between the two mixers enough to say that, well, this mixer is better than that mixer? So some difference will be there, but it will not be such a great difference that you can simply say, okay, this is good. Difference will be there, and based on that you have to take a decision. Further, I'll tell you one more situation where a decision will be difficult to take. So there will be a benefit, there will be a risk. So depending, this decision has to be taken based on benefit versus risk. In that type of situation, we have to take a decision, and that is where statistics is going to help us, particularly analysis of variance technique is going to help us. Now imagine a person in a managerial position from among 4 analytical procedures. So there are 4 different analytical procedures. You have to choose one. Or from among 3 surgical procedures, one surgeon is presented with 3 procedures. He has to select one. Or a manager is given 5 formulations and has to select one formulation, or 4 different equipments are there for mixing and we have to choose one. In all these situations, it is difficult to take a decision, and Anova is useful in all these situations. Further, I'll tell this to you. This Anova or the statistical procedure will not only help you in taking a decision, it will take a decision in a scientific manner. There will be a risk versus benefit analysis, and clearly Anova will help you. You will be able to tell right in your thesis or right in your research paper, because analysis of variance has declared that there is significant difference. A, B, I have used the multiple comparison test, and among the 4, this one is the best. This difference is clearly significant. That is why I have chosen this procedure. So it will give you a very systematic, scientific way of answering a question and taking a decision. Sorry. So when you test for the significance of difference between means of, say, 3 formulations by finding out their bioavailability, when each is tested on, say, 5 individuals, it is one-way Anova. So suppose you have tested some formulations, 3 formulations you have chosen, and you are making a comparison between their bioavailability. You have administered them to 5 individuals, then you are comparing. So how do you do the experiment? 5 volunteers, 1, 2, 3, 4 and 5 were asked to take formulation A on a day, and their plasma samples were collected, and AUC was determined from the plasma samples. So this is the way I am giving the rough protocol for the experiment also, for the bioavailability study also. So you take 5 volunteers, give them product A, and as per the protocol, collect their plasma samples, determine what is the amount of drug in the plasma samples, you submit it to analysis, and you get bioavailability. You draw the area under the curve and you get the area under the curve. Now the same volunteers will take the next B formulation after 1 week of washout period. Again you take plasma samples, you determine the bioavailability. In the 3rd week also you repeat the same. So now what are your results? Patient number 1, 2, 3, 4, 5, area under the curve from formulation A, area under the curve from B, area under the curve from C. So these are all indicating the bioavailability. Bioavailability, and remember the same 5 persons are taking A, B and C. Now what do? We wish to test 2 points, whether there is significant difference between the mean bioavailabilities and whether the bioavailability of these formulations is changing with populations of different groups. Then that is 2-way Anova. In 1-way Anova, now what do? You carry out 1-way analysis of variance. By the suitable procedure, which I am going to give you shortly, you will carry out analysis of variance, and you will be able to write here under this decision, yes, there is significant difference, or no, there is no significant difference. Like that you can write the decision and you can select. Now when you will go for 2-way Anova, suppose there are 2 points here. We want to test whether A, B, C and are different, and whether the bioavailability of these formulations is changing with populations of different age groups. We are checking for significance in 2 directions. It is 2-way Anova. Now how that is changing? How the experiment is changing? We categorize our test sample of volunteers into 4 age groups: 21 to 30, 31 to 40, 41 to 50, 51 to 60. So see, in this here we have taken only 5 people. All are similar people. In this 2-way Anova, what we have taken? 4 varieties of groups of patients we have taken, each group belonging to different age. We take 9 volunteers into each group. In this group 9, in this group 9, in this group 9 and in this group 9. In each group of 9, by lottery, we fix which 3 people get A, which 3 people get B and which 3 people get C. We administer the medicines, we take the blood samples, we determine the AUC. Now remember, the experimental protocol is still the same. In this 9 people, 21 to 30 age, 9 people, by lottery we fix which 3 people are going to get A, which 3 people B, which 3 people C. Similarly, we do in all the age groups. Now we do the Anova again. This is known as the Anova table. See, in this age group, 3 volunteers get A, 3 volunteers get B, 3 volunteers get C. And so this is how we prepare the table. In each place, we take the mean of the values that are coming and we carry out two-way analysis of variance by the procedure we are just going to learn in a few minutes. So, and then we'll have two decisions in our hands. We can declare, is the difference between A and B and C significant or not? We can declare whether the different age groups are different with respect to their response to formulation A, B and C. So this is what is known as two-way analysis of variance. In this direction and in this direction, in two directions, we are finding out whether variation is significant or not. Then what is three-way Anova? We want to check the difference of significance among means in 3 directions. We test for difference among 3 formulations A, B and C, among 3 age groups, and we carry out the analysis of variance in 3 different laboratories. Now, are the 3 formulations different in their AUCs? Are the different age groups different in their AUCs? Are the different labs different in the results they are giving for the 3 formulations? Like that. So, earlier we are giving in 2 directions. Now we will give in 3 directions. Is there any difference between the formulations? Is there any difference between the age groups? Is there any difference between the different labs in which it is going on? So it will be like this: laboratory 1, laboratory 2, laboratory 3. Age group. See, intentionally I have kept 3 on this side, 3 on this side and 3 formulations. That will be easy for the calculation. That's why I kept. So this type of design is also would also be known as the Latin square design. So here 3 formulations, 3, 1, 2, 3. So then we will be giving 3 decisions based on this type of Anova table. Or is there any difference between 3 formulations? Our original objective. Is there any difference between the different age groups? Second objective. Is there any difference between the 3 laboratories? The third objective. So 3 decisions will be able to give based on the 3-way analysis of variance. So each formulation is getting tested, each age group is being tested, each laboratory is being tested. We separate the effect of the treatments from the effects of the age and the laboratory by doing the analysis of variance. So this is how one-way Anova, two-way Anova and three-way Anova will help us in finding out whether a certain difference observed practically between 3 formulations or 3 age groups or 3 laboratories is significant or not. So let us see what is the principle of analysis of variance. Total variance of all the data in an experiment can be separated and attributed to 2 sources: variance between groups and variance within groups. So this is for one-way Anova. Between group variance is high or large, within group variance is usually less or small. So let me take a minute to explain this point to you, because this is the principle. This is very important. So friends, let us see, please understand something. Usually I am saying, in the experiment that you carry out on the table in your laboratory, whether it is the pharmaceutical chemistry lab or Sutics lab or analysis lab, when you do the experiment on the table using a burette and a pipette or a spectrophotometer or whatever it is, I mean laboratory experiments. In this type of experiments, the variance is really not so high. The variance is usually small. Suppose you are doing a titration, your teacher asks you to do a titration. The first time you do the titration, you may get some error. Second time you are nearer, more accurate. Third time, definitely accurate. Fourth time, definitely you will have a value which is consecutively agreeing. So that's what I mean. Error is less. Assuming that you are having reasonable skills, error is less when you are working in the laboratory. Unlike that, when you go on to do biological experimentation, when you are working with animals or human volunteers, the variation is quite large. Whatever you may do exactly the same procedure every time without making any difference. Still there is variation. This variation is because of intersubject variability and intrasubject variability. Different people or different animals are different. A same person or same animal is different on different days. As a result, variance is very high. That is the reason why we have to take into consideration the variation. So what does analysis of variance do? Analysis of variance will consider the total variance among the results of the sample, and that variation is or variance is divided into two categories. One category which is between groups, between different groups, and that is supposed to be large, that is usually high. Within a group is usually less. So the variance is divided into between groups and within groups, and what Anova does is it will form a ratio of this between groups variance to within group variance. If this ratio, this is between groups is taken in the numerator, within groups is taken in the denominator. If the ratio is very high, then we declare the difference to be significant. If the ratio is very less, we declare the variance to be not significant. This is the principle of Anova. So variance within group is also called as error variance. So just now I said variance within a group is small usually. That's why we call it as error. We also call it as the chance variance. I'll talk more about this as I go along this lecture. If the experimental treatment has been effective, the between groups differences will be expected to be greater than can be accounted for by chance. Suppose we are giving a treatment. What do we mean by a treatment? For example, in this experiment, what we are doing, let us go back to the first one. So here what we are doing, we are giving a formulation A to all the patients. So this is what we mean by treatment. By treatment, we mean that we are causing a change. We are giving a formulation. Formulation means a medicine. So the patient is consuming this medicine. So the medicine has gone into his blood. That blood value we are determining. So this is one treatment, second treatment, third treatment. So these are the treatments. Now, only these are the treatments. These are not treatments. These are called blocks. These are the population on which we are doing the experiment. Here what we are doing is, we are dividing, categorizing our population or the sample of patients who have come as volunteers into 4 blocks. Each block being different from the other. So these are called as blocks. These are called as treatments. When we give a medicine, that is called as a treatment. Then, so this is how these are blocks. These are treatments. These are called as columns. These are called as rows. These are called as treatments. So because we have given a treatment, there is a difference in the value. So if really the treatment is effective, the between group differences will be expected to be more than what can be accounted for by chance. Let me go back again to this. If really there is a treatment, there is a difference between A, B and C, then the difference between these formulations, whatever difference comes, variation comes between these, will be more than what is seen as per chance between these patients. So that is what we mean by seeing. Within groups is less than that. So this is how one-way analysis of variance we are showing by a pie chart. In a one-way analysis of variance, this entire circle is representing the total variance observed among all the people. So this entire blue thing is variance due to real reason because of the formulations ABC. This one is the variance due to chance reason. So 3 important assumptions are there in analysis of variance. The observations are independent. Present population from which observations are taken, parent population is normal. Various treatment and environmental factors are additive in nature. Now these are important assumptions. Observations are independent. One should not be dependent on the other. There should be no relation between the dependence on the other. And the parent population is normal. All these tests, Z test, T test, Anova, are parametric tests, and they have an underlying assumption that the parent population from which the observations are taken is normal, and these variances are additive in nature. So among all these, let us explain. One observation is not affected by the other. The result of AUC or bioavailability of one patient has nothing to do with the results of AUC or bioavailability of another patient. This is the meaning of the word. They are independent. So what is parent population is normal? The bell-shaped curve. If you take normal observations like height or weight or IQ of people of a region, they are all normal population. This is what we mean by a normal. All normal attributes like height of people, weight of people, etc., they follow a normal, means they follow a bell-shaped curve. If you take the values of the, let us say the weight of 1,000 people, then categorize them into different categories. Suppose the mean weight of the 1,000 people is 50. Then suppose the standard deviation is 10. Then 50 is here. So in between 40 and 60 kgs, 68.26% of the 1,000 people will lie. In between 30 and 70, 95.44% of the people will lie. And in between 20 and 80 kgs, 99.73% of the values will lie. This is so, this will always happen because the weight of people is a normal attribute. Unless you go into, so we always give this rider. Suppose I talked about taking 1,000 people and determining their weight. If you do this in any place, in a college or in a town or in a city, anywhere it is all right. Only thing is you should not go to a place where specifically obese people may be there or specifically lean people may be there. There you will not find this type of relationship, but normally as long as a normal population goes, this will be held. So this is what we mean by parent population is normal. So we assume that the effects of the treatment and other effects are additive in nature. The variances are additive in nature. Variance within a class among different classes. We are comparing the mean performance of 3 different sections of the same class. I wish to take another example which will be nearer to you because many of you must be students. Usually within the same section there is lesser variability, and between different sections there will be more variability. Let us suppose there is a college where third B Pharm or any B Pharm class is there. There are 3 sections of B Pharm class are there. B Pharm final year class, 3 sections are there. We conduct an examination for all the 3 sections. We find out their marks. So we have all the data, section A, section B, section C, and all the data pooled together we have. Now you will agree with me when I say that within one section there is lesser variance. Among all the sections there is more variance. This is because a particular section of class, section A, is exposed to the same type of teaching and same type of experience, whereas different sections may be exposed to different type of teachings. So the variance among different sections is expected to be more, and the variance within the section is expected to be less. This is what we mean. So another word, significance, I want to explain. Between different sections, more variability is expected. If the ratio between groups of variance, real error, and within groups, if the ratio is taken, if it is higher than the ratio recorded in F tables, we declare the difference to be significant. Otherwise, we say that it is not significant. Then there are what are known as F tables. I'll show you presently how an F table can be read. And in an F table, there will be a value that is known as the critical value. If we find out the ratio between groups variance, within group error, then if you take a ratio, if that ratio is more than the F value in the table, then that is supposed to be give us the significance. So the variability in the data is partitioned or divided into 2 parts. One is variability within a group, other is the variability between different groups. This is what we mean by partitioning. Another example I am taking. Suppose there are 3 tablet machines A, B and C. There is a production division.
Hello, my dear participants. It looks like a power cut problem. We will see for 2 minutes. I will talk to the speaker. So please be on hold. Thank you.
Hello.
Hello, hello, madam. Hello, madam. Is it audible, madam? Hello, Vijaya Ratna, ma'am.
Hello, Iswar. Am I audible?
Yes, ma'am. Yes, madam. Yes, ma'am. You can proceed, madam. Everything is fine. Yeah.
So we want to see whether there is significant difference between the tablets from the three machines when tested by some parameter such as hardness, weight, or dissolution time or disintegration time. So we operate the machines under identical conditions and we bring out identical type of tablets. From each we take 6 tablets in a random manner. We find out the we randomly pick them and we take the particular thing and we find out. So chance variance is, which is small, it is unavoidable and creeps into all experimental results due to pure chance reasons. So some variation will always be there. So from each machine, we are picking up 6 tablets and determining the dissolution time. So totally from each machine, 6 tablets, 18 tablets totally. So between the 3 machines also some difference will be there. Within the same machine also some difference will be there. You will not get 18 identical values, but the chance variance will be there usually within the same machine. This is unavoidable. All values will not exactly be identical. There will be small difference. This small difference is known as chance variance. This is unavoidable. And then the real variance will be there. Suppose one machine is clearly different from another machine in its efficiency, then there will be a definite variation and that will be real variance. This will be large. This is avoidable. So this is the reason why this is also called as the real variance. So another example I can give you, taking microcapsules. Suppose we take ethyl cellulose, methyl cellulose and cellulose acetate. This is what I have done for my PhD work. With 3 different polymers, I have taken a drug and we prepared the microcapsules. Then we observed, we found out the drug release from the different microcapsules and we found out that the difference is significant. By doing analysis of variance, we have found. So basically what I am telling is that what is analysis of variance? It is a technique which will help us make a decision with respect to is the variations observed in the results significant or not. Can we clearly say with confidence that this product is better than that product? So the purpose of Anova is to establish whether variation between groups is likely to be a function of chance or not. So here you see comparison of 3 batches of tablets with their dissolution times. So I think I have given enough examples initially with respect to 3 different treatments, then with respect to tablet machines, then with respect to microcapsules. Now let us go on and work out certain problems and see how the problems are to be worked out. See here, the dissolution times of the tablets taken from 3 different batches are seen. Is the difference significant or not? So partitioning sum of squares is there. So the basic ingredient of Anova is the sum of squares, SS. Sum of squares is a measure of variability. Total sum of squares, between group sum of squares and within group sum of squares are what we calculate. Total sum of squares is the sum of squares of the deviations of each of the observations from the grand mean. Please look here, you will understand. See, there are here how many values? From batch 1 there are 5 values, batch 2 there are 5 values, batch 3 there are 5 values. Within the same batch also there is some variation. Between the different batches also there is some variation. So what now we want to do? We want to find out the variance among all the 15 values and categorize that into 2 varieties. One is within batch, another is between batches. So how do we do that? I am giving you the concept. Listen. So initially we will find out a mean for all the values. Let us call this X double bar, grand mean of all the values. Then what do we do? The concept I am telling, remember. So for every value, we will subtract from the mean value. That may be a positive or a negative value. We make a square of that. We add up all those squares. That is the concept of the total variance. Total variance include the variance of each value from the grand mean value. Then the same concept we do for batch 1. We find out the mean of this and find out the difference between the single value and the batch mean. Then for the different batches also we do that. This is the process that goes on concept wise. Let us see. So sum of squares is the basic unit of analysis of variance. So this is the concept I just now talked about. So we find out the grand mean of the total sum and then take out every value from the grand mean, make a square of that and add up all those values. This is known as SST or total sum of squares. Then the sum of squares within groups is the sum of squares of the deviations of each individual score in each group from the mean of this group. So how is this done? See, suppose we take a mean of the first batch, some value will be there. Take out each value from that mean, make squares of all that, make a sum of all that. That is the concept of within group sum of squares. So this is how we do that. Concept of sum of squares of within groups. Sum of squares between groups is where from the grand mean, we take out the mean of each value and we multiply with that. Sum of squares between groups is obtained by taking the value from each mean and we doing that. So this is the concept of between group sum of squares. Then here total sum of squares is just now I told. But the formula what we do is total sum of squares is given by sigma xi square minus sigma x whole square by capital N, where SST is the total sum of squares, xi is the individual score, x bar t is the grand mean of all the values, total number of observations, and sigma x square is the sum of the squares of all the values. See, again I am going to tell you how to do the total sum of squares. Make a sum of all the values. That is the total sum. Sigma x. Make a square of that. Make a square of that. Make a square of the total sum. Divide by capital N. In this case, capital N is 15. 15 values are there. So that is also known as t square by n. So the first thing we calculate when we do Anova is t square by n. Make a sum of all the values, square and divide by n. Then make a square of each value and make a sum of that. Sigma x square. Sigma x square minus sigma x whole square by n will give you the total sum of squares. So this is what is the computational formula. Sigma x square minus sigma x whole square by n will give you the total sum of squares. Then sum of squares between groups is obtained by sigma x1 whole square by n1 plus sigma x2 whole square by n2 plus so on minus sigma x whole square by n. This last term, as you can see, is t square by n. Just what I told just now. Sum of all the values square divided by n. This is also known as the correction factor or t square by n and is used in every formula. Now, how do you get between group sum of squares? Again, let me go back to these values and explain to you. Make a sum of this batch and square it. Divide by 5. That is sigma x square by n. See, this is the computational formula. Like that you do for all the 3 batches and subtract the t square by n. Sum of a batch, square, divide by number in the batch. So in Anova, you can have different numbers in different batches also. That is also possible. So for some within group sum of squares, this big formula is there, but the computational formula is very simple. We calculate the total sum of squares and subtract the between sum of squares from that to get the within group sum of squares. So and finally we write what is known as the schematic. This is the table that we write. See, treatments are given by k minus 1, error is given by n minus k, total is given by n minus 1. This is known as the degrees of freedom. Let me explain the concept of degrees of freedom to you. The degrees of freedom has to do with the maneuverability or the capacity to change, capacity to alter the values that are there in that. So roughly, I'll give you this example. What is actually given in physical pharmacy, Martin, I suppose. Suppose there is a junction having 4 roads. 4 roads are there going from a junction. Suppose one car enters from one road. Then as it reaches the junction, you can predict that it will take any one of 3 roads. Then let that car be there. Then another car enters. Now there is already one car in the one road. Suppose we say this car can only take a road which other car has not taken. Then one car is there already. So the second car has only 2 and a third car has only 1, like that. How much variability you have, capacity to maneuverability you have. I'll tell another more simple example. Suppose we are saying that there are 4 values. The sum of the values is 100. You can only fix 3 values. Once you fix the 3 values, the 4th value is already fixed. So then when there are 4 values, the degrees of freedom is 3. That's how the concept of degrees of freedom is there, k minus 1, etc. So the sum of squares is just now what I talked about. And when you divide the sum of squares by the degrees of freedom, you get the mean sum of squares. And when you take the ratio of mean between sum of squares by mean within group sum of squares, you get the F ratio. Then when you go to the F ratio table, then when you compare with the table value, you can make the decision. Then, so for the given problem, then the degrees of freedom are 2, error is 12, total is 14. These are the sum of squares and this is the F ratio. And because the F ratio is more than the table value, we reject the null hypothesis and we say that the 3 batches are different. Now, there is another point here that I wish to explain to you as they are doing analysis of variance. Look here, the values are 77, 81 and so on. So you have to square these values, you have to make a sum of that. Instead of that, there is a ease of way of doing in analysis of variance where you can subtract a value from all these values so that the numbers will become very simple, and do calculations with those numbers. Calculation is very easy. So you can subtract 70 from all these. So that is known as coding. You can subtract the same number from all the values. So look here. So what we have done, coding by subtracting 70. So all these values are there. Please see that there is a minus value here. When there is minus, you take that as minus. When you make a sum of these, it will be 2 plus 4 is 6. This is minus 16. So the total will be plus minus 10. Like that, including the minus sign, you make a sum. You must remember that. Otherwise, this is known as coding. So this is the book from which you can take the tables. Pharmaceutical Statistics by Bolton is the book which I used for preparing this material as well as for taking the tables. So look at this. This is the table I told you about. F distribution, upper 5% values of the F distribution. So how do we look at the F value? So here in this table, see the F value is 8.47 practical value. So the critical value is to be obtained. How do you get the critical value? F2, 12. 2 is the numerator degrees of freedom. 12 is the denominator degrees of freedom. So here, see degrees of freedom in numerator, degrees of freedom in denominator. So 2 and 12. So what is the F value, table F value? 3.89. So because our practical value is 8.47, which is more than the table value of 3.89, we reject the null hypothesis. That is how we do the problem. Now, there is a testing procedure for analysis of variance, which is the same one which you use for t-test, f-test or Anova or any variety of test. Here, first of all, we take a null hypothesis where we say mu1 is equal to mu2 is equal to mu3. That is, all the samples are taken from the same type of population and the means of are all equal. And the alternative hypothesis says mu's are not all equal. So, writing of this procedure, doing of this procedure is very important from an examination point of view. Every step has a mark. If you don't write the null hypothesis, certain marks will be cut. So you have to write everything. This will also help you understand the problem. And you have to write all this before you do the calculation. So then the level of significance is 0.05. So the level of significance has to do with the probability of acceptance of our value. So here also you will find in this table it is, see here 5%. That means this is true for 0.05%. There will be another table for 0.01. So at a certain level of significance, the value is taken. Then we have to write the criterion statement. Reject the null hypothesis if f is greater than 3.89. The value of f 0.05, this is how we have to write. For k minus 1 is 3 and n minus k is 12 degrees of freedom, where f is to be determined by an analysis of variance. Otherwise, accept it. So, if f is greater than 3.89, reject. If it is less than 3.89, accept. Reject what? Accept what? It's null hypothesis. Null hypothesis to be rejected if our practical f value is more than 3.89. Null hypothesis to be accepted if the calculated value is less than 3.89. This is the criterion statement that we write. Then the calculations are done like this. Total sum of squares is obtained by sigma xi square minus sigma xi whole square by n. SSB, just now I explained these formulas to you. SSW is obtained by total sum of squares minus between group sum of squares. Then we write the Anova table. Then we take the decision. If f value is obtained is more than that, reject H0, otherwise accept it. In our case, we got an f value of 8.47, which is more than 3.89. So we reject the hypothesis. We take the decision that the 3 batches of the tablet are different. That's how we are doing the analysis of variance. So just now I have explained to you the procedure for the one-way analysis of variance. So in two-way Anova, as I explained earlier, there are rows, there are columns, there are error. Now the variance is divided into 3 components. The error, that the variance that is there in the rows, the variance that is there in the columns, the variance that is there in the because of the error. So in the two-way Anova, 3 varieties of variance is there, and this is how the schematic is different. So treatments, blocks, error, total. So we are calculating 2 F ratios. So because of treatments, earlier example, please remember, I told about 3 treatments, giving medicine A, B, C, 3 treatments, and blocks, 4 age groups. Those are blocks. Blocks are the difference that is there in the blocks, the variance that is there in the treatments, then the variance because of the chance reason. So you calculate 2 F ratios and you take 2 decisions. Then, so here I am giving another example. Suppose you select a group of patients, divide them, categorize them into old patients, young patients, children and infants, and you take the drug also in 3 doses, low dose, medium dose, high dose, and 3 different treatments, and this is how you get the values. These are the mean values. Remember earlier I told you 3 patients, 3 patients, 3 patients. So these are average values that we are taking, mean values. And how do you do the degrees of freedom? Same variety. Treatments are, how many treatments are there? 3 treatments are there. So the degrees of freedom will be 2 actually. Here, this has, these 2 are intermixed. So the blocks are, treatments are actually 2 and blocks are actually 3. So you determine the sum of squares. By dividing, you get the mean sum of squares. Then you get the F ratios and you compare with the table values. In this procedure, there will be 2 table values. With those 2 table values, you can make a comparison. So here you see, I am giving another example where we take male and female rats are given the same dose of a hypnotic drug. So in two-way Anova, we are making a comparison here between male and female rats, and this is actually one-way Anova. We are making a comparison between male and female rats, and we calculate in one-way Anova total sum of squares, within group sum of squares, and between group sum of squares. Then we write the table. See, the source of variation is between groups, within groups. Total sum of squares, degrees of freedom, mean sum of squares, and the F ratio is 9.14. So F19 table value is 5.32. So first column, 8th row. Let us verify whether this is right or not. Go back to the F table given in Bolton. So 1 and 8 is 5.32. 5.32 is the table value. As a result, then we can, because this value is more than 5.32, we reject and we say that there is a difference between female rats and male rats, and female rats sleep longer than male rats. So analysis of variance has helped us in making this decision. Observe that here we have taken only, this is a comparison between only two. So that further reinforces one more point to you. In some situations, especially in biological experiments, even when we are making comparison between two mean values, we use analysis of variance. Analysis of variance is a very versatile type of technique, and this is how we use it. So here we are taking two drugs, two-way Anova, two male and female rats, drug A and drug B. We are making a two-way analysis of variance. See, we make the calculations like this and we form the table. So between columns, between rows, and within groups sum of squares. And for each we determine the table value and we make a decision. So when tables and columns are there, it is easier for us to say that rows and columns rather than blocks and treatments. So rows and columns we make. In three-way Anova, we are compartmentalizing the variation into 4 groups: rows, columns, and errors. So here you see treatments are having maximum effect. Next rows, next columns, next error variance is very, very low. So from this, we could make 4 conclusions. Females sleep longer than males. Drug A has a longer duration. Drug A causes especially long sleep in females or especially brief sleep in males. So here, what we are able to do is we are able to get the interaction effect also. So when you do the calculations in a certain manner, two-way analysis of variance is also helping us in finding out the interaction effect. So in three-way Anova, we find out total sum of squares, row sum of squares, column sum of squares, treatment sum of squares, and error sum of squares is obtained by making a sum of rows plus columns plus treatments and subtracting that from the total sum of squares. So here we do the calculations in a similar manner, and only thing I want to tell you here is, so the column sum of squares, you add up all the columns, row sum of squares, you add up all the rows. Treatments, how do you do? A here, A in the second column is here, A in the third column is here, A in the fourth column is here. So for the treatment A, what is the sum? 3 plus 2 is 5 plus 3 is 8 plus 4 is 12. So this is how you do the three-way analysis of variance. So here there are 3 null hypotheses. Here the total sum of squares is obtained in the same manner. Calculations are exactly same. And the three-way Anova schematic is like this. You take 3 decisions, and we deliberately we keep all the rows are equal to columns are equal to treatments. So this variety of Anova is useful in Latin sum of square Latin square design. So we take 3 decisions here. So now I want to move on to another concept and tell you about the use of multiple comparison tests. Okay, come here. You have found that rows are different between columns or treatment between treatments. But which row is different, which column is different? Specifically, let me talk about the research of one of my scholars, RDLP Christian, and tell you in that context you will understand better. So what is multiple comparison? If we get the answer in Anova that the difference is among is significant, then the next logical step would be to ascertain between which 2 means difference is significant and between which 2 means it is not significant. We calculate a mean significant difference by some formula or method, and if the difference between 2 means is more than that mean significant difference, we declare it is significant. Otherwise, we say that it is not significant. So suppose one researcher has worked with 4 plants. From the 4 plants he has done ANOVA, he found the difference to be significant. Now, which 2 plants are different? Which 2 plants are not different? To find out that answer, we have to do multiple comparison. Several varieties are there. I'll tell you about 2 tests, Skiff's method and Dunnett's test, in terms of what my student has done. In Skiff's method for multiple comparison, we compare a number of treatments. We also show significance for shorter intervals than some other tests. It is also known as the post hoc test for ANOVA. It is named by the American statistician. So, in all these tests, what is the mechanism we do? We find out what is known as the least significant difference, LSD, least significant difference. So, we calculate a particular difference which is above which we say it is significant, below which we say it is not significant. Next step, what we do? What is the difference between the means of each one? Treatment 1, treatment 2, treatment 3, treatment 4, we find out and we make a comparison with the LSD. If it is more than the LSD, we say yes, this is significant. If it is less than the LSD, we say no, it is not significant. So this is the formula by Steps method, S square into K minus 1 into F into 1 by N1 plus 1 by N2, the entire thing under square root. So S square is the appropriate variance estimate. Within means sum space for one way ANOVA. K is the number of treatments and N1 and N2 are the sizes of the 2 samples being compared. F is the table value of F from the F table with degrees of freedom K minus 1. And again, we obtain a value using this equation. If the practical difference between the means is more than this value, we declare that it is significant at the given alpha level. In Dunnett's test, we make a comparison taking a control. When there is a control and comparison, then we use the Dunnett's test. Most of you are research scholars, especially in the field of pharmacognosy or pharmacology, you will know what control means, or in clinical pharmacy. We usually have a control where we don't give any treatment. It's like our blank. So when there is a control and treatment, then we use Dunnett's test. So the table that I am talking about, Dunnett's test table, is taken from Bolton book. So this is the Dunnett's test from which we take the table. In a given experiment, A may be control and B and C and D may be treatments. In this type of situation, we do Dunnett's test, where we take the formula for Dunnett's test. D is given by this formula. The value of T is obtained from standard tables. So I said I will talk from the angle of my student's work. One of my student has worked with some plants. Those plants are having nootropic activity. So he took out the extracts of those plants. He administered those extracts to animals and find out there are 2 experiments I am talking about. So he administered the extracts and he observed with a photooctometer how the activity has improved. In other words, how the memory has improved. With respect to that, he compared his results with ANOVA. He found that there is a significant difference, and then he went on to do multiple comparison tests to find out which 2 plants are significantly different. Let us see that. So this is the results of my student. So he compared with phenytoin. You know, phenytoin is a drug which is used in epilepsy. So phenytoin alone he has given, that is one treatment. Piracetam is the standard drug to be used in the case of people who are with respect to memory. So this is one plant, another aqueous extract, alcoholic extract, aqueous extract, like this. So how many total? 8 treatments are there. So all these are different doses are there. So he found the mean activity, he has made a comparison. So by looking at the values, you have certain idea. For example, piracetam is 91. So this is 38, this is 42, this is 50, like that. So I think as the thing is coming down, it is supposed to be more effective, like that. So the control versus treatment. Control is this. These are treatments. These are AB, AC, AD. This is the difference between A and B. 20.8 and 91.2 difference is this. Then A and C. 20.8 and 38, the difference is minus 17. Like this, he found the differences, and he found the differences to be very high, significant. Then BC also difference to be very significant. Here he has given between group sum of squares, within group sum of squares, F ratio is very, very, very high, highly significant. So significance is there. So statistical by way of ANOVA followed by Schiff's method for multiple comparisons is what he has done, and he found that all the treatments are very, very, very effective. So this is how he found the difference to be highly significant. So then the alcoholic extracts of the drugs were found to be dose dependent, and the effect was found to be very, very significant. Further, then what he did, he gave these extract to the animals. At the end of certain period, the animals were sacrificed, and from the brains of the rats, the acetylcholinesterase activity was found out. So he did this work on dosage forms. So please remember my opening statements. I was trying to tell you that this will help you in optimization. This will help you in making decisions with respect to your formulations. So he prepared 3 or 4 formulations and tested them by the nootropic activity. And this is the example for the Dunnett's test. So control group, standard drug, formulation 1, formulation 2, formulation 3. These are syrups made from the herbal extracts. So dose is vehicle. This is the activity 8, 4, 6, 5 and 4. So one-way ANOVA is highly significant. So data is expressed as this. So statistical analysis by ANOVA followed by Dunnett's test. So here you are seeing control is this, standard is this, and all 3 are showing good activity. So with the help of the Dunnett's test, he could find that there is a significant difference between which one he found. So here I want to emphasize once again. So one-way ANOVA is telling us that there is significant difference between the control and all these. So how is the Dunnett's test helping us? Dunnett's test is helping us in establishing between which 2 plants, between which 2 formulations there is significant difference. If there is no significant difference between 2, it will tell. So in this case, in all the cases, there is significant difference that has come. So the help of the multiple comparison test with the help of Schiff's test or Dunnett's test is they will help us in determining after ANOVA which 2 plants or which 2 formulations are significantly different. That is where these are helping. So we'll move on to another thing. So this I am very much eager to tell you that actually in the design of experiments also this ANOVA will come, and by understanding ANOVA and its implications and its interpretation, you are able to do your DOE better. Where will you do your DOE? In your optimization technique by quality of design. So in optimization techniques, we select experimental points so that the entire area of interest is covered. We separate the effects of the variables. We can tell which variable caused a particular result. So how do we do this? Suppose one of you, a research scholar, you want to prepare a matrix tablet. For that matrix tablet, you have to choose the polymer you have chosen, diluent you have chosen. What is the weight of the polymer? What is the weight of the diluent you want to choose? So that you can prepare tablets of a particular hardness or a particular 100% dissolution time. So what is the optimization procedure I am telling you? First of all, we do some preliminary experimentation and determine the range of the 2 excipients to get the dependent variables as we want them. So what is this? With the polymer and the diluent taking different weights roughly, we prepare a set of experiments, say 10 experiments. Those results we feed into the design of experiments. We select a design of experiments, maybe central composite design. We feed into the template of the design. Then the design will tell us, you carry on. So we'll tell the design. So this is the range of the results. I want a particular hardness. I want 4 kgs per square centimeters. I want 100% dissolution in 80 minutes. You give the design of experiments, these things, the results of your preliminary experimentation and the Y variable you want. Then the central experiment, the central composite design will give you certain things and will ask you to do the trials. Then we'll carry out all those experiments and give our results into the software. Then the design will give us certain plots, and these plots help us to do the optimization. So these varieties of plots will be there. This is known as response surface methodology. I am giving these which were made by another scholar who has prepared tamsulosin hydrochloride loaded neosomes. Now this plot and this plot will help you pick up the values of the independent variables of the polymer and the diluent, so that you can have a tablet of the given hardness and dissolution time. So this is what is known as optimization or design of experiments. In this, what is the role of analysis of variance? So this is the type of analysis of variance table that it will give you. Let me go through this once again. Just now I told. So for a given model, so in getting the design, after you got the results, you want to know whether the results that you got, which are given here, are following a linear model or an interactive model or a quadratic model. So for each model, it will give you an ANOVA table. Then it will give you a sum of squares. Sum of squares, as I told you, is the total sum of squares and so on. Degrees of freedom, mean sum of squares. Sum of squares divided by degrees of freedom is giving you the mean sum of squares. The F ratio is obtained by dividing this mean sum of squares with the residual sum of squares you are getting. So you will get the F value and the P value, and significant means which is significant, and it will give you the lack of fit and the total value also. So based upon this ANOVA table, you will know which polymer, which variable is significant, which variable is not significant. This lack of fit is also important. It will tell you when you are comparing the linear, interactive and quadratic model. Based upon this lack of fit value, the significance of the lack of fit, you will decide which model is most significant. Whether your data is fitting the linear model or quadratic model, you will make a decision. So this is how, this is the normal plot of residuals. This is the residual sum of squares. So this residual sum of squares is like the error sum of squares, which has come from this. So this is the mean value. This line indicates the mean. See, every point is deviating from the mean. So if you take the difference of this point from the mean, make a square of that and make squares of all and make a sum, that is known as the residual sum of squares. That is like the error sum of squares. So these are the terms in the ANOVA which I have already explained to you. So where F is more, there we reject, otherwise we accept. Here I have also explained the probability to you. So then coming to my conclusions, I would like to explain what I have done. I would like to summarize what I said by this. So I try to emphasize that analysis of variance is a statistical tool which will help you take decisions. It will help you take decisions in a number of situations. And the multiple comparison test will help you take a decision with respect to which two treatments the difference is there. And analysis of variance is a big part in quality of design, in design of experiments, which will help you make a decision with respect to which product, fit is better. Your data is fitting linear or which model it is fitting. So that's how analysis of variance is a very versatile technique used in a number of situations and useful in a number of ways. So here I have given the references: Bolton and a book by Richard A. Johnson and probability, research work of my scholar RDLP Christian, and Stephanie Glynn, a website which I have seen in taking certain things. So I hope I have reached you. Hope you have got a better understanding of ANOVA now than earlier. I thank the organizers for giving me this opportunity, and I thank you for your patient hearing. Thank you all once again.
Hello, ma'am.
Yes.
That's really a wonderful session. I went back to my college days because I am proud to say that I am your student. So your energy levels and all that.
I am relieved, Iswar. I was worried because of the technical. You understood really well?
No, yes, man, everything is fine. With just 1 or 2 minutes, technical glitches are quite common. So, and we need to accept when we want good stuff. So don't worry about that, ma'am. It went very well. and your interest or your what I can say depth of knowledge is there. It is clearly visible from your presentation. So we will take some of the questions from the audience now.
Gaurav Kole, what is washout period? Why is it essentially required in Anova? Washout period, dear student, has to do with the bioavailability study part, not Anova. When we give one treatment to a particular patient, for 1 week it will be there in his blood. So we give a washout period of 1 week. 1 week later only we give another treatment. So it has to do with the bioavailability study, not Anova. Satyasri, good morning, madam. What is the minimum sample size required for three-way ANOVA? Three-way ANOVA means there will be columns, rows, and also treatments. Usually, 3 is very, very minimum. For more than 3, it should be there. More than 3 rows should be there. More than 3 columns should be there. More than 3 treatments should be there. Singaravalam Sindhuja, good morning. The analysis of disease progression using same samples between control and disease, what would be preferred, one-way or two-way ANOVA? Control and disease. Only 2 samples are there, control patients and treatment patients. One-way ANOVA only, one-way only. Only when there is one angle of comparison, one-way ANOVA only. Taragesh, should both correlation and variance analysis results by statistically significant? In both we are talking about significance. Yes, in correlation also we have to tell whether it is significant or not. Yes.
Yes, ma'am. So as the time is running short, we will close the question and answer session. So now I request Dr. R. Vijay, faculty at CL Baid Metha College of Pharmacy, to give the concluding remarks for the second session by Dr. J. Vijaya Ratna.
I would like to thank Professor J. Vijaya Ratna ma'am. It was well explained elaborately and keenly by using practical situations in an easy, accessible way, like a one-way, ANOVA, two-way, ANOVA, multi-comparison method. By giving this tool method, Schiff's method and Gentle's method, it was extensively clarified the implications of GOE also, ma'am. Thank you very much, ma'am, for being with us for this webinar. Thank you, ma'am.
So now we have our 2 speakers on board. Hello, Ravi sir. How did you felt about the second session?
Thank you, thank you. It was a very nice session, ma'am. Thank you, ma'am.
Thank you.
So now I request Dr. C.N. Nalini ma'am to give her remarks on the second session. Ma'am, please unmute your mic, ma'am.
It was an excellent presentation. All the examples given were so simple that the students could easily understand. It was very nice session, ma'am.
Thank you.
Yeah. Sir, Vijay Kumar sir. Hello, Vijay Kumar sir. One second, sir. I am checking with Vijay Kumar, sir.
Yes, I'm there.
Hello, Vijay Kumar, sir,
yeah.
Is it audible?
Yes, yeah, audible.
You please turn the mobile, sir. It is showing in
vertical
way. Very good. Yeah. Sir, now may I request you to please give the vote of thanks for today's Series 8.
Yeah, it's my pleasure to myself, R. Vijay Kumar, Assistant Professor, CL Baid Metha College of Pharmacy, and it's a pleasure to proceed with vote of thanks here on behalf of our college, CL Baid Metha College of Pharmacy. I'd like to thank our chairman, secretary, and management of management staffs for the technical support and every step of our success. And I extend my gratitude to our beloved principal, Professor Dr. Grace Ratnam, for her greatness in guidance and for the successful completion of this webinar series. And I thank Ravi Jayapal sir, who readily accepted our invitation in his busy schedule. He was there with us for more than 2 or nearly 3 hours with us. And I thank our second speaker, Professor Vijaya Ratna madam. And she is more energetic. And when you say retired professor, and it is not looking like a retired professor, I thank her also with her energetical way to giving a speech in that way. And sir, Ravi sir, given current practices, whatever going on in industry, we are seeing looking like we are inside of industry. At how you given a lot of impurities profiles and how we are minimizing it, how we are going to tackle with that. And I thank Ravi sir and also our second speaker, Vijaya Ratna madam, very heartfelt thanks. And I thank our HOD, Dr. C.N. Nalini madam, who given wonderful suggestions in that conducting of webinar, and our staff, Dr. Amudalakshmi madam, Dr. Vijay madam, for their cooperation in this entire session to successful completion. I thank Dr. Ubaidullah and Dr. Priyanka madam for the technical support in this webinar series. And not only technical support, I thank our staff Sivakumar and Satish Kumar for their technical support in association with MYBO group especially. This would have good in presenting in YouTube. Very good, excellent presentation what we have in the screen now. In future also we do in conditions like this. And I thank especially the Ishwar sir from MYBO group and other staffs who are in this MYBO group who are supporting this conducting of webinar series. And not least, I thank all our participants who registered in this webinar series and getting enlightened speeches from our excellent speakers, and continue with the webinar series. I thank all of you taking part in this webinar series. Who are not missed, I thank them also. Suppose not mentioning them, don't feel, and all are here. I am thanking to whoever taken part and behind the screen and on the screen. I thank all of you to getting a part in the thank you very much. Thank you, sir.
Thank you. Thank you.
So, yeah, that is really a very good session we had with two excellent speakers, one from California, USA, and another from Visakhapatnam, India. So I hope all the participants enjoyed both the sessions because both are very interesting and both are highly required in research as well as in industry perspective. So the topic selections are also excellent by the organizer, CL Baid Metha College of Pharmacy. On behalf of MYBO group, I would like to thank Dr. Grace Ratnam madam for giving us an opportunity to get associated with the very famous and oldest college in India, that is CL Baid Metha College of Pharmacy. For that, we are feeling very much privileged, madam. Thank you very much. And I thank all the faculty members, particularly Dr. C.N. Nalini ma'am, Dr. S. Amudalakshmi ma'am, Dr. N. Vijay ma'am, and Vijay Kumar sir. And I especially thank Dr. U. Ubaidullah sir, Dr. Satish Kumar sir, and Mr. R. Shivakumar sir, who are what I can say, instrumental in making MYBO group associate with CL Baid Metha College of Pharmacy. So thank you one and all for giving the opportunity. I take it as a privilege. So we hope we will continue our journey. So thank you one and all. And now it's time to sign off. Thank you one. So all the certificate links and feedback links will be mailed to the registered email IDs to all the participants. No need to worry. So we'll be in touch with you through email of registered mail IDs. Thank you one and all. Thank you Ravi sir. Thank you Vijaya Ratna ma'am and everyone. Bye bye.